Welcome to Bedtime Science. Did you know that your blood contains thousands of proteins that slowly change as you age? And that some of them, found in abundance when you are young, may hold the key to reversing the aging of your brain? A research team at Stanford, led by Tony Wiscore, discovered that roughly 3,000 proteins circulate through your bloodstream at this very moment. From a single blood sample, their algorithm can estimate your biological age within a couple of years. Not your calendar age, your real age, the age your cells are actually living. Tonight we look at 100 quiet facts about how the human body ages, what it needs to thrive, and how the things we eat shape the years we live. In the next moments, we will gently discuss how aging happens not in a slow fade but in sudden molecular bursts, with concentrated waves of change arriving in the early 40s and again in the early 60s. We will look at why your tongue is wired as a survival instrument, with 5 hardwired taste qualities, each one designed to keep you alive. We will examine how factors circulating in young blood can rejuvenate memory and reactivate dormant stem cells in the aging brain. And we will consider what the rate of puberty reveals about the speed of your biological clock, and what tiny chemical tags on your DNA can predict about lifespan. Before we begin, if you find comfort here tonight, consider subscribing to join our quiet community. And if you are still awake, you can leave a soft comment about what topic we should explore next. Now let's begin. Your blood is a river of information. It carries oxygen, it carries nutrients, but it also carries something less obvious: thousands of proteins, each one secreted by tissues throughout your body, each one circulating at concentrations that shift predictably as you grow older. These proteins aren't decorative. They influence how your cells behave, how your organs communicate with one another, and how quickly your body moves through time. Tony Wiscore and his colleagues at Stanford analyzed plasma drawn from 4,263 people, ranging in age from 18 to 95. They measured thousands of circulating proteins in each sample, and what they found reshaped the way scientists think about the biology of growing old. Roughly 1/3 of all proteins measured changed markedly with advancing age. Your blood isn't a static pool. It's a living, dynamic molecular record, updated with every heartbeat, revised year after year after year. From this proteomic data, they built a predictive aging clock. It could estimate an individual's chronological age within approximately 3 years, using nothing but the composite levels of proteins found in a single blood draw. Think about that. 3 years of accuracy from a tube of blood. That's a precision comparable to simply asking someone how old they are, except the blood sometimes tells a different story than the birthday on your driver's license. A separate team built an even more refined version. They trained a machine learning model on data from 3,301 individuals and achieved a Pearson correlation of 0.98 in the training set and 0.96 in the test set, with a median error of just 1.84 years. Less than 2 years of uncertainty. From a mathematical reading of proteins. It's among the most accurate biological age predictors ever assembled. Here's the thing. That same clock showed something with real practical weight. People who exercised regularly, specifically aerobic exercise, had a measurably younger predicted biological age than sedentary individuals of the same calendar age. Your habits show up in your blood. The proteins remember what you did this week. Wiscore's group then extended this work into territory no one had mapped before. They built organ-specific biological age models using nearly 5,000 plasma proteins measured across 5,676 individuals in 5 separate cohorts. They mapped age predictions to 11 distinct organs: adipose tissue, artery, brain, heart, immune system, intestine, kidney, liver, lung, muscle, and pancreas. 11 organs, each with its own biological clock, each ticking at its own pace inside the very same body. And what they discovered was that your organs don't age together. Not at all. In their study, roughly 18.4% of individuals showed what they called an extreme agiotype, a profile dominated by accelerated aging in one single organ. Your heart might be 5 years older than the rest of you. Your kidneys might be younger. Only about 1.7% showed extreme accelerated aging across multiple organs at once. For most people, aging is uneven, patchy. It arrives in different rooms of the body at different times, like water slowly finding its way through an old building. Disease leaves traces in these organ clocks. People with hypertension showed kidneys approximately 1 year biologically older than their peers without hypertension. People with diabetes showed kidneys about 1.3 years older. Atrial fibrillation was linked to a heart that appeared 2.8 years older than expected. A prior heart attack pushed that number to 2.6 years of accelerated cardiac aging. And each standard deviation increase in heart biological age, roughly equivalent to about 4.1 additional years of heart aging, conferred an almost 2.5-fold increased risk of heart failure over a 15-year follow-up. That connection between a blood-based biomarker and future organ failure is one of the strongest ever documented in prospective research. Your blood knows what your heart is going through before you feel a single symptom. A large-scale replication study confirmed all of this, applying organ-specific aging models trained on 2,916 proteins from 44,498 individuals in the UK Biobank. The results held. Plasma proteomics could estimate biological age across all 11 organ systems and predict future disease onset and mortality. The clinical translation potential is real. It sits in the proteins passing through your veins right now as you listen. Let your breath slow for a moment. These clocks are ticking. They don't make a sound. There's another clock running alongside the protomic one. This one is written not in proteins, but in tiny chemical tags attached to your DNA. In 2013, Steve Horvath at UCLA developed the first pan-tissue epigenetic clock. He used methylation levels at exactly 353 specific sites in the genome, places where a small chemical group called a methyl group either sits or doesn't sit on the DNA strand, to predict biological age across 51 different human tissue types and cell types. Horvath trained it on 8,000 samples from 82 datasets, and one of the most striking findings was this: DNA methylation age is close to 0 in embryonic and induced pluripotent stem cells. Reprogramming a cell to its earliest state essentially resets this clock. The methylation tags begin accumulating from birth. They start immediately, and they keep accumulating steadily for as long as you live. Consider this. All 20 cancer types examined using the Horvath clock displayed epigenetic age acceleration relative to healthy tissue. The average acceleration was 36 years. Cancer cells don't just grow wrong. They age wrong. They resemble tissue that is more than 3 decades older than the person carrying them. More refined clocks followed in the years after. GrimAge was designed not to estimate how old you are, but to predict mortality risk. It consistently outperforms earlier clocks in predicting time to death and age-related disease onset across large populations. Dunedin pace, which stands for pace of aging computed from the epigenome, was designed to measure something different still. Not how old you are biologically, but how fast you're aging right now. That distinction matters. It means Dunedin pace can detect whether something you're doing today, this month, this season, is actually slowing or accelerating your biological clock. Faster biological aging, as measured by the epigenome, predicts shorter lifespan and earlier mortality. And the rate of physical maturation during puberty appears to correlate with the speed of long-term aging. Those who mature faster may age faster over the full arc of their lives. The clock starts early. It remembers everything. Among the proteins that anchor these organ-specific aging models are names you might not recognize, but whose roles are quietly profound. NPPB and TNNT2 track heart aging. Renin and uromodulin measure kidney aging. Clotho, a protein named after a figure from Greek mythology who spun the thread of life, serves as a systemic longevity factor. Complexin 1 and 2, along with stathmin 2, act as signatures of neurological aging. Each one a molecular proxy for tissue-specific decline, readable from a single tube of blood drawn from your arm on any given morning. These first observations describe the clocks that measure aging, but aging itself doesn't happen the way most people imagine. For most of the 20th century, aging was pictured as a smooth decline, a slow dimming, a gradual fade from young to old, like a candle burning steadily down. But a landmark study published in 2024 in Nature Aging dismantled that image entirely. Researchers tracked more than 135,000 distinct molecules in 108 healthy volunteers, aged 25 to 75. These volunteers gave biological samples every 3 to 6 months over a median period of 1.7 years. The molecules measured included RNA, proteins, metabolites, lipids, and constituents of the gut microbiome. The analysis generated nearly 250 billion individual data points. 250 billion. It was one of the most data-dense longitudinal studies of human molecular aging ever attempted. And what it revealed was this. Rather than changing gradually, 81% of the molecules studied showed nonlinear fluctuations across the lifespan. Not a slow drift, not a steady decline, a series of step changes and surges, concentrated at specific ages. Two distinct crests of molecular change appeared. They arrived at average ages of approximately 44 and 60. Michael Snyder, the senior study author and Stanford's W. Asherman Professor in Genetics, described it plainly. We're not just changing gradually over time. There are some really dramatic changes. It turns out the mid-40s is a time of dramatic change, as is the early 60s. And that's true no matter what class of molecules you look at. During the mid-40s wave, the molecular pathways most affected included alcohol metabolism, lipid metabolism, caffeine metabolism, cardiovascular signaling, and markers linked to skin and muscle composition. If you've ever noticed that hangovers hit harder after 40, or that injuries take longer to heal, or that your relationship with a morning cup of coffee has quietly shifted, there are molecular reasons for all of it. The 40s aren't just a calendar milestone. They're a biological event. During the early 60s wave, the most prominent changes appeared in carbohydrate metabolism, immune regulation, including shifts in inflammatory cytokine levels, and kidney function. This corresponds precisely to the clinically recognized surge in risk for diabetes, infections, and renal disease that begins in the seventh decade. The body isn't just older, it has undergone a concentrated molecular restructuring. Cardiovascular disease-related molecules showed changes at both peaks, both at 44 and at 60, suggesting that the cardiovascular system goes through two distinct windows of accelerated molecular remodeling across adult life. Not one late-life collapse, two separate waves, decades apart. An earlier protomic study by Wiscore's group, looking at 4,263 participants, had already identified 3 inflection points in protein-level aging at approximate ages 34, 60, and 78. The nonlinear pattern shows up across multiple independent datasets and methods. Aging comes in waves. Think of it like tides, not a river, not a clock running down. Tides that rise, hold, and then pull back before rising again. These observations about how aging arrives in bursts are drawn from what the blood reveals. But blood isn't only a readout, it is an active participant. The circulatory system doesn't just measure aging, it shapes it. Here's where the story takes a turn that might surprise you. One of the most quietly provocative discoveries in modern biology began with a 19th century surgical technique called parabiosis. Two mice, one young and one old, are joined surgically so that they share a continuous circulatory system. The blood of the young mouse flows through the old mouse's body, and the old mouse's blood flows through the young. When old mice were joined to young partners, something happened in the older mouse's brain. Hippocampal learning and memory improved. Brain inflammation decreased. Neurogenesis, the birth of new brain cells, increased. Young blood, it turned out, contained systemic factors capable of reversing aspects of brain aging. Young blood exposure also increased the density of dendritic spines on mature neurons in the hippocampus of aged mice and improved synaptic plasticity, the ability of neurons to strengthen or weaken their connections in response to experience. These were structural changes, not just behavioral improvements. The old mice's brains were physically rebuilding connections that had been lost. Part of this cognitive enhancement was traced to a specific molecular pathway. Exposure to young blood activated a transcription factor called CREB in the aged hippocampus. CREB regulates the expression of genes involved in synaptic strengthening and memory consolidation. The young blood was turning on the brain's own repair instructions. The code was already there. It just needed a signal to run. And it wasn't just the surgical joining that mattered. A single blood exchange between old and young mice, without shared organs, produced similar rejuvenating outcomes. Even more precisely, plasma transfer experiments, using only the cell-free fraction of blood with no cells at all, showed that soluble, protein-based factors were the primary agents. Something dissolved in the fluid itself was doing the work. Scientists began identifying what those factors were. CCL11, also called eotaxin, is a chemokine whose circulating levels increase with age. It was identified as a pro-aging factor. It activates immune cells called microglia in the hippocampus, disrupts synaptic connections, and impairs cognitive function. When old mice received young bone marrow transplants and their CCL11 levels dropped, their hippocampal learning and memory were preserved. Removing the bad signal helped nearly as much as adding a good one. On the rejuvenating side, two proteins were pinpointed: THBS4, thrombospondin 4, and SBARCL1, SBARC-like1, were identified as synaptogenic factors enriched in young plasma. They promote the formation of new synaptic connections in aged brain tissue. They help the old brain build new bridges between neurons, the way a city might lay fresh cable along streets that had gone dark. GDF11, or growth differentiation factor 11, had a more complicated history. It was initially reported as a rejuvenating circulating factor that reversed age-related cardiac hypertrophy and improved skeletal muscle and brain function in old mice. Subsequent studies contested the heart findings, but exogenous GDF11 has since been shown to improve brain vasculature, increase neural stem cell numbers, and remodel aged blood vessels. An incomplete picture, but a compelling one. But here's what matters most from these experiments. Dormant stem cells in the aged brain can be reactivated by exposure to young blood. The limiting factor in neurogenesis, in the brain's ability to grow new neurons, isn't the exhaustion of the stem cell pool itself. The stem cells are still there, waiting. They've gone quiet, but the inhibitory systemic environment created by aging blood is suppressing them. Change the blood, and they wake up. Your body already carries stem cells in your brain that are capable of renewal. They're just listening to the wrong signals. There's a protein released during exercise that speaks directly to the brain. It's called irisin. It's cleaved from a membrane precursor called FNDC5 during physical activity, and it crosses the blood-brain barrier. Once inside the brain, it activates a signaling pathway that induces the transcription of BDNF, brain-derived neurotrophic factor, along with other neuroprotective genes in the hippocampus. Physical exercise becomes, through irisin, a form of systemic blood-borne brain protection. And the evidence is specific. Circulating irisin rescues memory impairment in Alzheimer's disease mouse models. When researchers blocked either peripheral or brain-derived irisin, the improvements in synaptic plasticity and memory normally produced by exercise vanished completely. Irisin isn't just one of many signals. It's the critical intermediary. Without it, exercise doesn't reach the brain the same way. Based on all of these parabiasis and plasma transfer findings, Tony Wiscore initiated clinical trials in which patients with Alzheimer's disease received infusions of young plasma, typically representing 5 to 10% of blood volume. Preliminary results have generated cautious optimism. The idea sounds like something from a novel, but it's quiet biology. Your blood isn't just fuel, it's a broadcast network. And the message it carries, young or old, inflamed or calm, reaches every organ with each heartbeat. In older men, each standard deviation increase in circulating interleukin-6, or IL-6, is associated with a 15% decrease in survival time and a 0.94-year reduction in lifespan. Each standard deviation increase in C-reactive protein, or CRP, is associated with a 12% decrease in survival time and a 1-year reduction in lifespan. These two inflammatory markers are among the most reliable predictors of how long a person will live. Simple blood tests, quiet numbers, they tell the story of your immune system's relationship with time. Let that settle for a moment. Your circulatory system is a communication channel, whispering instructions to every tissue it touches. The next group of facts turns to something closer to your daily experience, the sense of taste, specifically, how the tongue functions as one of the oldest survival instruments in the human body. Charles Zucker's laboratory at Columbia University's Zuckerman Institute has spent 2 decades mapping the molecular and neural architecture of taste. What they've revealed is a system far more precise and far more ancient than most people realize. The mammalian taste system is organized around exactly 5 fundamental taste qualities: sweet, salty, umami, bitter, and sour. Each one is detected by a dedicated population of taste receptor cells on the tongue and palate. Each one transmits signals along dedicated neural pathways to corresponding brain regions. There's no blending at the level of the receptor cell. Each cell knows its job. Sweet, umami, and low-concentration salt are classified by the nervous system as appetitive. They're innately rewarding. They evolved to guide you toward calorie-dense foods, protein sources, and essential electrolytes. When your tongue detects sweetness, your brain receives a signal that means energy. When it detects umami, the taste of glutamate and nucleotides in protein-rich foods, the signal means amino acids, building blocks, survival material. Bitter and sour are classified as aversive, innately repellent. Bitter taste specifically expanded in evolutionary diversity during the transition from aquatic to terrestrial life. On land, plants presented vertebrates with a novel chemical defense system of alkaloids, tannins, glycosides, and hundreds of other toxins. The bitter receptor system expanded to meet that threat. In amphibians, bitter taste receptor genes range from 39 to 178 distinct variants per species. That's a vast detection array, tuned for one purpose: don't eat the poison. Zucker described the taste system's logic with a musical analogy. He said you can think of the palate like a piano with 5 basic keys. The combination of keys and the intensity with which the piano is played transforms the chemicals detected on the tongue into flavors you either enjoy or reject. 5 keys, but an endless range of compositions. Each of the 5 taste receptor cell types is narrowly tuned. The vast majority of taste ganglion neurons respond strongly to only one taste quality. This is called a labeled line architecture. It preserves categorical purity from the tongue all the way to the brain. Sweet stays sweet, bitter stays bitter. The signals don't mix on the way up. Look, here's something Zucker's team discovered about salt in 2013. Extremely salty foods activate not only salt-sensing cells, but also the aversive neural pathways for sour and bitter. That's why a small amount of salt tastes good, but a mouthful of pure salt makes you recoil. The aversion isn't a preference. It's wired into the circuitry. Your brain treats too much salt the way it treats a toxin. In a landmark 2015 paper published in Nature, Zucker's team demonstrated something even more striking. By selectively activating or silencing specific clusters of neurons in the mouse gustatory cortex, they could make mice behave as if they were tasting sweet or bitter, without any chemical stimulus on the tongue at all. The tongue wasn't involved. The brain generated the taste experience by itself. Taste perception, at its deepest level, is a brain-generated construct. The tongue is a sensor. The brain is the interpreter, and the interpreter can be activated without the sensor even being consulted. Taste receptor cells are among the most rapidly replaced sensory cells in your body. Each individual taste cell lives for just 10 days to approximately 6 weeks. The entire population of taste cells within every taste bud undergoes complete renewal roughly every 2 months. Right now, you are tasting the world with cells that didn't exist 2 months ago. And yet you have no conscious awareness of this replacement. The brain maintains its expectation of taste quality even as the hardware detecting it is continuously rebuilt. A seamless handoff, happening beneath your awareness over and over for your entire life. But aging disrupts the regenerative capacity of this system. In young rodents, complete functional recovery from gustatory nerve injury takes approximately 40 to 45 days. In aged rodents, the same recovery extends well past 50 to 65 days, more than double the recovery time. Minimal taste bud regeneration was observed even at the later time points. This delayed regeneration was accompanied by impaired neural-immune interactions in the tongue lining. The immune system and the taste system talk to each other. When the immune system ages and slows down, a process called immunosenescence, it takes the taste system down with it. This offers a molecular explanation for the common experience among older adults that food tastes blander. It's not just perception. The tongue is literally slower to rebuild itself. Children's aversion to bitter vegetables, the broccoli refusal, the Brussels sprout negotiation at dinner is not stubbornness. It's an evolved safety mechanism. Bitterness signals plant toxins. A child's nervous system is calibrated to reject those signals with maximum force, but neuroplasticity allows this aversion to shift across the lifespan. Adults learn to accept and even enjoy bitter flavors. Coffee, dark chocolate, arugula. The wiring changes. The survival system gradually loosens its grip. In 2025, Zucker and his Columbia laboratory unveiled the full three-dimensional structure of the human sweet taste receptor. This discovery was originally enabled by the lab's identification of the sweet receptor gene back in 2001. It opens the door to rational drug design aimed at modulating sweetness perception without adding sugar. If you can make the brain perceive sweetness without the caloric cost, you change the equation between pleasure and survival. Let your jaw relax. Let your tongue rest softly behind your teeth. That small muscle has been keeping you alive since before you were born. These facts about taste describe the body's front gate, the first line of chemical assessment between you and the world of food. The observations that follow go deeper into what happens after the food enters and how the body builds itself from what it receives. There's a common assumption that muscle-building nutrition works differently in men and women, but research by Laura Colenso-Semple and others has shown that muscle protein synthesis in response to nutrition is remarkably similar between the sexes. The cellular machinery that builds muscle doesn't distinguish between chromosomes. The major sex difference in muscle is hormonal. Testosterone levels drive size differences. But the fundamental response of muscle cell to amino acids and exercise is shared. Leucin. It's the branch-chain amino acid most abundant in animal proteins, and it's the most potent activator of a signaling complex called mTORC1. This is the cellular switch that tells your body, build. Leucin increases the activity of downstream targets approximately threefold more than any other essential amino acid at equivalent concentrations. When leucin arrives, the cell gets the message. Protein synthesis should begin. The primary molecular sensor for leucin is a protein called leucyl T RNA synthetase, or LARS. It binds directly to a GT pace called RAG in a leucin-dependent manner, and this binding activates the mTORC1 signaling cascade. There's a second sensing mechanism too. A stress-inducible protein called cestrin-2 acts as an intracellular leucin sensor. In the absence of leucin, cestrin-2 inhibits mTORC1 by holding onto a complex called GATOR2. When leucin binds to cestrin-2, the inhibition is released and mTORC1 activation proceeds. 2 locks, 1 key. But as the body ages, something goes wrong with this system. It's called anabolic resistance, the blunted muscle protein synthesis response to amino acid and exercise stimuli that develops over time. It's a major driver of age-related sarcopenia, the gradual loss of muscle mass and strength. In older individuals, mTORC1 signaling can remain suppressed for up to 24 hours after exercise. In younger individuals, the signal fires and fades much faster. The machinery still works, but the volume knob has been turned way down. And honestly, here's where the paradox gets interesting. mTORC1 activation is essential for building and maintaining muscle. You need it. Without it, you lose mass, you lose strength, you lose the ability to stand up from a chair without thinking about it. But chronic pharmacological inhibition of mTORC1 with a drug called rapamycin consistently extends lifespan in mice. The same pathway that builds muscle also accelerates systemic aging when left running without pause. Rapamycin-mediated inhibition of the downstream kinase S6K1 delays the onset of multiple age-related problems in mice: bone matrix loss, immune dysfunction, motor dysfunction, insulin resistance. These effects are independent of lifespan extension itself. mTOR is a master regulator of the rate of aging across multiple organ systems. It's the gas pedal. Press it hard to build, ease off to last. Growth and longevity pull in opposite directions. The body has to negotiate between them every single day. Your food contains molecules that quietly modulate these pathways with every meal. Sulforaphane, an isothiocyanate compound found in cruciferous vegetables, especially broccoli sprouts, functions as an epigenetic modifier. It inhibits an enzyme called DNA methyltransferase 1, reducing the methylation of a promoter region that controls a master transcription factor called NRF2. When NRF2 expression increases, it activates an entire program of cellular antioxidant and detoxification enzymes. One compound from a handful of broccoli sprouts, reaching into the epigenetic machinery of your cells. Sulforaphane also functions as a histone deacetylase inhibitor. It simultaneously reduces the activity of 4 separate HDAC enzymes: HDAC1, HDAC4, HDAC5, and HDAC7, while suppressing multiple DNA methyltransferases. It's one of the few food-derived compounds capable of modulating both DNA methylation and histone acetylation in the same cell. 2 layers of epigenetic control, influenced by a single molecule you might eat in a salad or a stir-fry. Resveratrol, the polyphenol found in red wine and grape skins, activates a protein called SIRT1, sirtuin-1. SIRT1 then promotes mitochondrial biogenesis through a transcriptional coactivator called PGC1-alpha. This cascade enhances mitochondrial function, increases fatty acid oxidation, and improves cellular energy balance in ways that mimic the metabolic effects of eating less. A glass of wine doesn't make you immortal, but the resveratrol in it taps into the same pathways that fasting activates. Resveratrol also induces autophagy through a pathway involving AMPK, ACT, and mTOR. It triggers both the canonical and non-canonical forms of autophagy, removing damaged organelles and misfolded proteins that would otherwise pile up and accelerate cellular aging. Quercetin, a flavonoid polyphenol found in onions, apples, and capers, promotes autophagy through activation of TFEB, transcription factor EB, the master regulator of lysosomal biogenesis. It increases the number and capacity of the cellular garbage disposal apparatus, more lysosomes, better clearance, a cleaner cellular interior. Polyphenols broadly delay cellular aging by reducing oxidative stress and preserving telomere stability. The molecular mechanisms include epigenetic upregulation of HTERT, the gene for human telomerase reverse transcriptase, via SIRT1, and the support of mitochondrial biogenesis through AMPK activation. These aren't miracle compounds. They're the quiet chemical signals embedded in colorful fruits and vegetables, signals that whisper to your cells, slow down, clean up, repair. There's another molecule that declines with age in a way that cascades through nearly every system in your body. NAD+, nicotinamide adenine dinucleotide. Its levels drop approximately twofold in multiple tissues, including liver and skeletal muscle, between youth and middle age. This systemic decline has been described as a cascade of robustness breakdown. As NAD+ falls, the sirtuins that depend on it lose their activity. Mitochondrial function deteriorates. DNA repair slows. The entire maintenance system begins to falter, like a building where the custodial staff has been slowly reduced year after year. Administration of NMN, nicotinamide mononucleotide, a direct precursor to NAD+, restores NAD+ levels in aged tissues and reverses multiple hallmarks of aging in mice. Mitochondrial dysfunction improves. Age-related inflammation in adipose tissue decreases. Insulin secretion and action are enhanced. Neuronal function gets better. Another precursor, nicotinamide riboside or NR, follows a similar pathway through a conversion step via NR kinase before entering NAD+ synthesis, and confers overlapping metabolic benefits. The food you eat isn't just fuel. It's a set of molecular instructions delivered to your cells with each bite, shaping the speed at which your body moves through time. The next observations turn to the gut, specifically to the trillions of microorganisms living inside your digestive tract, and what they reveal about the biology of an exceptionally long life. The gut microbiome isn't a passive bystander in digestion. It's a metabolically active organ whose composition shifts profoundly with age, and its character in people who live past 100 differs in measurable ways from that of younger individuals. Centenarian gut microbiomes show enrichment of specific bacterial species, not typically dominant in younger populations. Among them is Akkermansia muciniphila, a gram-negative anaerobe that degrades mucin in the gut lining to produce acetate and propionate. Also present in elevated amounts, Christensenella minuta and various lactobacillus species, including Lactobacillus plantarum. These organisms aren't random passengers. They're associated with protection against inflammatory bowel disease, metabolic syndromes, obesity, and type 2 diabetes. The bacteria in a centenarian's gut look different because they're doing different work. Short-chain fatty acids, principally butyrate, acetate, and propionate, are the microbial fermentation products that maintain the gut barrier, fuel the cells lining the colon, and exert systemic anti-inflammatory effects. SCFA levels decline with advancing age in most people, but they're detectably higher in centenarians than in age-matched elderly controls. The centenarian gut is still producing the molecules that keep inflammation at bay. Butyrate specifically reinforces gut barrier integrity by promoting the expression of tight junction proteins. It reduces intestinal permeability, the leakiness that allows bacterial products to escape into the bloodstream and drive chronic inflammation. And it inhibits NF-kappa B signaling, one of the master switches for inflammatory gene expression. One metabolite, produced by bacteria in your gut, doing the quiet work of holding your intestinal wall together. Italian centenarian studies have identified elevated circulating levels of two distinctive microbial metabolites, phenylacetyl-glutamine and p-cresol sulfate, as signatures of the centenarian microbiome. These are produced from aromatic amino acids by specific fermentative bacteria that are largely absent or less active in younger populations. The chemistry of extreme old age has a microbial signature, written in compounds most people have never heard of, produced by organisms too small to see. These observations about the microbiome connect to a broader pattern. The gut feeds the immune system. The immune system shapes the inflammatory environment, and the inflammatory environment determines how fast your organs age. The following facts move to what happens when light reaches your body, and what happens when food doesn't arrive for a while. Every cell in your body is capable of responding to light if light can reach it. In the skin, sunlight activates melanocytes in the epidermis. Genetic programs switch on to produce pigmentation. The process is automatic, coordinated, ancient. But deep organs, the spleen, the liver, the kidneys, remain in perpetual darkness. Light reaches the skin, the retina, and a few millimeters below the surface. Everything else lives in a world without photons. Your relationship with light extends well beyond pigmentation. Vitamin D synthesis begins when ultraviolet B radiation strikes 7-dehydrocholesterol in the skin, converting it into provitamin D3. This molecule is then slowly transformed in the liver and kidneys into its active form, calcitriol, which supports immune function, bone metabolism, and gene expression across dozens of tissue types. The sun doesn't just warm you. It initiates a biochemical cascade that reaches your bones, your immune cells, your DNA. When food stops arriving, even temporarily, the body activates a different kind of program. Fasting triggers autophagy. That word comes from the Greek for self-feeding. It's the cell's quiet recycling system. During autophagy, damaged proteins and worn-out organelles are enclosed in membrane-bound structures, delivered to lysosomes, and broken down into raw materials that the cell can reuse. Housekeeping at the molecular level. And it accelerates when the body senses a shortage of incoming nutrients. The timing of meals matters. Circadian alignment, eating during daylight hours and fasting during darkness, appears to enhance the body's metabolic efficiency. Time-restricted eating windows that match the body's natural circadian rhythms have shown benefits in animal models for metabolic health, weight regulation, and inflammatory markers. Your body doesn't just care about what you eat, it cares about when. Polyphenols from plant foods activate cellular stress pathways that promote repair. Sulforaphane upregulates NRF2. Resveratrol activates SIRT1 and AMPK. Quercetin promotes lysosomal biogenesis. These compounds create a mild cellular stress, a phenomenon called hormesis, that triggers the body's own defense and repair systems. A little stress from the right source makes the system stronger, like exercise for your cells. Vitamin D's relationship to longevity extends beyond bone health. Low vitamin D status has been associated with increased risk of cardiovascular disease, autoimmune conditions, cognitive decline, and all-cause mortality in large population studies. The body needs light, It needs the chemical conversation that begins when a photon of ultraviolet light strikes the surface of your skin. Let the image rest there for a moment. Light arriving on skin, a chemical conversation beginning, your body quietly converting sunlight into the molecules that protect your bones, your immune cells, your aging brain. The immune system reaches into nearly every corner of your health. It evolved primarily to protect against infection, but its influence on aging is profound and pervasive, winding through nearly every process we've discussed tonight. The facts that follow look at how the immune system ages and what that aging means for everything else. The thymus. It's a small organ behind your breastbone. It's the primary site where T cells are educated. It's where immature T cells learn to distinguish self from non-self, friendly from dangerous. But the thymus doesn't last in its original form. It begins to shrink in early adulthood. The functional tissue is gradually replaced by fat. The number of new, naive T cells entering your peripheral immune system drops with each passing decade. A transcription factor called FOXN1 is critical for maintaining the thymus. Its expression declines with age, driving what scientists call thymic involution. But here's the hopeful part. In experimental settings, restoring FOXN1 expression in aged mice partially reversed thymic regression and restored naive T cell output. The thymus doesn't die. It goes quiet, and under the right molecular signals, it can be coaxed back. There's direct human evidence for the importance of ongoing thymic function. Young adults who underwent thymectomy in early childhood, typically for congenital heart disease surgery, display premature immunosenescence compared to age-matched adults who kept their thymus. Their T-cell profiles resemble those of elderly individuals. The thymus isn't just a childhood organ. Its output is needed throughout an entire life. As T cells age, some of them stop dividing but don't die. They become senescent. And senescent T cells develop what's called the senescence-associated secretory phenotype, or SASP. They secrete pro-inflammatory cytokines, chemokines, and growth factors. These molecules amplify systemic inflammation and accelerate the senescence of neighboring cells. Old immune cells don't just stop working. They actively make the environment worse for the cells around them. Like a broken furnace that fills the house with smoke, instead of simply going cold. The practical consequences are measurable and striking. Only 30 to 40% of older adults mount an efficient immune response to influenza vaccination, compared to roughly 90% of young adults. The reduced naive T cell repertoire, the accumulation of senescent memory T cells, the impaired innate immune signaling, all of it contributes. The same shot enters the same muscle. A weaker response comes back. Alexander Marson at UCSF and the Gladstone Institutes has developed efficient CRISPR genome engineering tools specifically for primary human T cells. His laboratory can now systematically interrogate thousands of genetic loci that control T cell function, a platform that has revealed novel regulators of anti-tumor immunity, autoimmunity, and infection resistance. Using CRISPR-based gene silencing across 15,000 sites in the DNA surrounding the FOXP3 gene, which is essential for the development of regulatory T cells that prevent autoimmunity, Marson's team created the first complete functional map of the entire FOXP3 regulatory control system. They identified enhancers and a previously unknown repressor element. The instruction manual for immune self-tolerance, read in its entirety for the first time. In a 2025 study published in Nature Biotechnology, researchers at the Arc Institute, Gladstone, and UCSF used CRISPR-Raf and CRISPR-Ron, epigenetic editing tools that modify gene expression without cutting the DNA, to simultaneously silence and reactivate multiple genes in primary human T cells. They achieved high cell survival rates while creating enhanced CAR T cells with improved tumor control in mouse leukemia models. Gene editing without scissors. Epigenetic reprogramming of the immune system. Genome-wide CRISPR knockout screens in human T cells within tumors identified a protein called GNAS, a central signaling mediator downstream of multiple receptors that suppress T cell activity in the tumor microenvironment. When GNAS was knocked out, T cells became resistant to multiple suppressive signals and survival improved across diverse solid tumor models. One gene removed, and the immune cells could see the cancer again. No intervention alters the pace of biological aging quite as profoundly as the chronic disruption of circadian timing. Your body's relationship with darkness and rest shapes every molecular clock we've discussed tonight. The brain has its own waste clearance system. It's called the glymphatic system. It uses the spaces surrounding cerebral arteries and veins as channels through which cerebrospinal fluid flows, exchanging with brain interstitial fluid and sweeping soluble waste proteins into the lymphatic drainage system. It's the brain's plumbing, and it works best when you rest deeply. Glymphatic influx increases approximately 95% during slow-wave rest relative to wakefulness. During deep rest, the interstitial fluid space expands from about 20% of brain tissue volume to approximately 40%, nearly doubling the channel capacity available for waste transport. The brain literally opens up, creating wider channels for the cleaning fluid to flow through. Amyloid beta, the peptide whose accumulation into plaques is a hallmark of Alzheimer's disease, is cleared from the brain approximately twofold faster during rest than during wakefulness. Every night of deep rest is a night of amyloid clearance. Every night of disrupted rest is a night when the plaques have a chance to accumulate. Two publications in Nature from early 2024 demonstrated that synchronized neuronal firing is the master trigger for glymphatic waste clearance. When researchers blocked neuronal firing, waste clearance stopped entirely. The neurons that fire in coordinated patterns during deep rest are the ones that drive the cleaning process. The memorable phrase that emerged from this finding, neurons that fire together, shower together. But glymphatic transport efficiency declines with aging. Reduced polarization of a water channel protein called aquaporin-4 in astrocyte in feet, progressive enlargement of the perivascular space, accumulation of extracellular matrix proteins that obstruct fluid exchange, all of it contributes. Aging impairs waste clearance. Impaired clearance leads to protein accumulation, and protein accumulation further impairs clearance, a feedback loop. And deep rest is the primary force that pushes back against it. In the aged brain, the downstream drainage route for glymphatic outflow, the meningeal lymphatic vessels, shows progressive deterioration. CD4 plus and CD8 plus T cell populations expand in the meninges, driving chronic neuroinflammation and further narrowing the brain's exit routes for waste. The immune system, aging alongside the brain, makes the drainage problem worse. Insomnia accelerates biological aging as measured by grim-age and skin-blood clock epigenetic clocks. People with clinically diagnosed insomnia show acceleration of both clocks and a measurable reduction in DNA methylation-predicted telomere length compared to good restorers. Disrupted nights don't just leave you tired. They advance your molecular age. Epigenome-wide studies of insomnia patients reveal a global pattern of DNA hypomethylation, affecting 785,200 CpG sites across the genome. That pattern matches the hypomethylation seen in advanced chronological aging. At the level of the epigenome, insomnia looks like fast-forwarding through time. Worsening rest quality, measured across domains including latency, duration, efficiency, and daytime dysfunction, is associated with accelerated aging on Grim Age and Dunedin pace. But not all epigenetic clocks respond equally to rest quality. Different clocks capture different biological processes. The relationship is real but layered, complex in ways scientists are still sorting out. Circadian disruption, the misalignment of your internal clock with the external light-dark cycle, has consequences that extend far beyond grogginess. Mice that lack a functional core circadian clock gene called BMAL1 exhibit dramatically shortened lifespans along with sarcopenia, cataracts, and multiple organ problems. Mice with normal internal clocks but exposed to artificially shortened light-dark cycles, 4 hours of light followed by 4 hours of dark, over and over, also show reduced lifespan. The mismatch between internal rhythm and external schedule accelerates aging on its own. And the evidence for the causal role of the circadian pacemaker is striking. Transplantation of fetal suprachiasmatic nucleus tissue, the brain's master circadian clock, into aged animals restores circadian rhythmicity and measurably extends lifespan. The degradation of circadian function isn't just a symptom of aging, it's a cause. Even subtle light at night carries risk. Chronic exposure to just 5 lux, approximately the level of a dim night light, for 24 weeks in aged female mice disrupted immune responses and reduced lifespan compared to females housed in complete darkness at night. The soft glow of a charging phone, the faint light from a hallway. In an aged system, even that can make a difference. But there is a counterweight. Caloric restriction rescues age-related decline in circadian metabolic pathways. It restores the amplitude and phase coherence of circadian gene expression networks, including SIRT1, NAMPT, and AMPK pathways that control the coupling between circadian rhythm and metabolism. These pathways lose their rhythmicity with normal aging. Caloric restriction brings the rhythm back. The metronome starts swinging again. Let yourself settle with these facts. Your body's relationship with light and darkness isn't a metaphor. It's a biological conversation happening in every cell, synchronized by a clock in your brain that has been keeping time since you first opened your eyes. The final facts in this collection step back from cells and molecules and look at something harder to measure, but no less real: the influence of human connection on the biology of aging. Among the most surprising findings in modern aging science is the extent to which psychological and social experiences leave measurable traces in the molecular machinery of aging. Not metaphorical traces, molecular ones, readable in the epigenome, in telomere length, in organ-level biological age estimates. In the UK Biobank study, one of the largest prospective cohort studies in existence, higher levels of both loneliness and social isolation were associated with accelerated biological aging. The effect was visible across multiple organ systems and epigenetic clock measures. Being alone doesn't just feel bad, it ages you. The cells know. And it isn't just the absence of connection that matters. Negative social ties, relationships defined by conflict, betrayal, hostility rather than warmth, have been identified as emerging risk factors for accelerated epigenetic aging. The quality of your relationships shapes the chemistry of your cells. A hostile relationship may age you faster than solitude itself. Loneliness is associated with shorter telomere length. A large longitudinal study measured telomere length 24 years after the loneliness measurement and found a moderate to large association. The biological embedding of social disconnection begins well before its clinical effects appear. It accumulates over decades, quietly, without symptoms, until the telomeres tell the story. The 5 blue zones, geographic pockets where exceptional longevity was first documented by demographers, offer converging natural experiments in which culture, diet, physical activity, and social structure combine to produce populations with extraordinary health spans. They are Okinawa, Japan, Sardinia, Italy, the Nicoya Peninsula of Costa Rica, Icaria, Greece, and Loma Linda, California. Each one is characterized by predominantly plant-based diets, moderate caloric intake, regular low-intensity physical activity, strong social cohesion, and a sense of purpose. Okinawans traditionally practice a principle called hara hachibu, the Confucian teaching of eating until approximately 80% full. It's a cultural caloric restriction practice that reduces oxidative stress, limits insulin secretion, and slows metabolic aging by creating a chronic mild energy deficit. It mirrors the effects of laboratory caloric restriction in animal models. Not a diet, a philosophy, encoded in a phrase repeated before every meal. In Sardinia, diets center on goat milk, pecorino cheese, Canonau wine, and high legume intake. The island has the highest concentration of male centenarians globally. On the Nicoya Peninsula, the traditional diet of corn, beans, and squash, sometimes called the Three Sisters, sustains a population where men have 2.2 years of greater life expectancy and 7 times the probability of reaching 100 compared to Japanese males. In Icaria, the Mediterranean diet combines with regular fasting aligned to the Greek Orthodox calendar, and the island has among the lowest dementia rates anywhere in the world. In Loma Linda, the Seventh-day Adventist community follows a plant-based diet with regular nut consumption, and its members live 7 to 10 years longer than the average Californian. The CALERIE trial, the comprehensive assessment of long-term effects of reducing intake of energy, was the first randomized controlled trial of caloric restriction in healthy, non-obese humans. Participants reduced their caloric intake by 25% over 2 years. The results were measured not in how people felt, but in how their molecular clocks responded. Caloric restriction slowed the pace of aging as measured by the Dunedin Pace epigenetic clock. It reduced circulating biomarkers of cellular senescence. It reduced fasting insulin and core body temperature, both biomarkers that are also reduced in long-lived animal models across evolutionarily distant species. These metabolic signatures appear to reflect a conserved biological program for extended lifespan. From worms to mice to humans, the same signals: eat less, age slower. Telomere attrition quantifies lifestyle damage with unsettling precision. Smoking 1 pack of cigarettes per day for 40 years is equivalent to 7.4 years of telomere-defined biological life lost. Obesity is associated with 8.8 years of equivalent telomere shortening. But here's what you should know. A lifestyle intervention incorporating physical activity, with or without dietary changes, can actually reverse telomere attrition. A meta-analysis of 20 studies involving 2,995 participants showed that intervention groups had a statistically marked increase in telomere length, while control groups declined. The damage is measurable, but so is the repair. Return now to where we started. The protomic clock, 3,000 proteins circulating in your blood, each one shifting in concentration as the years pass. A single blood draw, read by an algorithm, telling a story of biological age with a precision of less than 2 years. But the most vivid finding from all of this research, the one that stays with you, comes from the parabiosis work. Scientists took factors from young human blood and injected them into aging mouse brains. And what happened was quiet. No dramatic reversal. No sudden transformation. But dormant stem cells in the brain of those mice got reactivated. There was less inflammation, more activity, and most importantly, their memory function improved. As if the brain remembered how to be young. Tony Wiscore described the observation directly. There's stem cells in the brain of these mice that they got reactivated. There was less inflammation, more activity, and most importantly, their memory function improved. This is not science fiction. It's quiet biology. The body already contains the instructions for renewal. They're written in the proteins that circulate through every organ with each heartbeat. The challenge isn't inventing something new. It's understanding the signals that are already there and finding out which ones have gone quiet and why. The body ages in waves, not straight lines. Within the blood, within the food you choose, within the light that reaches your skin, there are quiet signals that shape how long and how well you live. Each meal is a set of molecular instructions. Each night of deep rest is a cleaning cycle for the brain. Each hour of sunlight is a chemical conversation between your skin and your bones and your immune system. Every meal is a small conversation between the body and time. Every connection with another person, every real, honest, unhurried moment of human contact leaves a trace in your telomeres, in your inflammatory markers, in the pace at which your epigenetic clocks advance. Every night of uninterrupted rest is your brain's opportunity to flush the waste that accumulated during the day, to consolidate what you learned, to rebuild what was damaged, to slow the quiet molecular changes that proceed whenever the cleaning system doesn't have time to finish its work. The science doesn't promise anything as simple as living forever. What it offers is something more modest and perhaps more useful: the ability to slow the clock, to preserve the function of your organs for longer, to compress the period of late life decline into the shortest arc possible. Not more years of aging, more years of living. Right now, as you rest, your body is quietly at work, repairing tissue, clearing old proteins, consolidating everything you learned today into memory. The slow clock of your biology turns gently forward. The proteins in your blood drift through every organ, carrying instructions written in concentrations you can't feel, but that shape everything about how your body moves through time. Your gut bacteria are fermenting the last traces of today's food into short-chain fatty acids that will hold your intestinal wall together through the night. Your thymus, however small it has become, is still sending out a trickle of naive T cells to patrol for threats you'll never notice. The methylation tags on your DNA have shifted, by fractions too small to feel, in response to everything you ate, everyone you spoke to, every hour of light and darkness you experience today. And the glymphatic system is opening now. The channels between your neurons are widening. Cerebrospinal fluid is beginning its slow sweep through your brain, carrying away the molecular debris of a day lived fully. The stars outside are not moving any faster than they were yesterday. The cells inside your body are not rushing anywhere. Everything is quiet. Everything is working. And rest, as it always has, carries you through. Sweet dreams, and we'll see you next time on Bedtime Science.